Hello, everyone. I am Carlos Munoz Amezcua. I work for Wolfram Alpha in biological content development, and I'm also finishing my master's program on computational biology and bioinformatics. Today, I will talk about making sense of biodiversity data with Wolfram language. In this talk, we will explore how Wolfram language can be used to make sense of complex structured data sets. Using a Darwin Core Archive of global jumping spider records as an example, we will use built-in tools for importing, inspecting, and visualizing structure in the data to inform the next steps in building a formal processing pipeline. A quick heads up, I will be talking about spiders and I will show a few pictures of them during this talk. So if you feel uncomfortable, you might want to consider the rest of this talk. Having said this, the spiders I will talk about are harmless and, in my opinion, quite charismatic as spiders go. I will start with a quick explanation about the context for you to better understand why I decided to use this dataset and what it is for. The dataset I will use in this talk was created for a paper published with a couple of colleagues and with a goal to describe how machine learning could be used as a tool in studying a special type of mimicry called partial mimicry. In this type of mimicry, the mimic resembles the target only at certain angles, typically because the mimicry occurs in parts of the mimic's body. For example, here on the left photo, if you kind of squint, you might see what looks like the face of a jumping spider with its legs, which you can compare with the real jumping spider on the right. Measuring this mimicry is an entire topic of itself. So without going too deep into it, I'll tell you that there are a few different methods. The one we set out to use in the study was to train a neural network to classify images of spiders versus other insects, and then challenging this trained network with images of mimics at different angles. So the results were interesting. So if you want to take a look at the paper, you can click on the title here. It will take you to the article. It's behind a paywall, so if you really want to read it and you cannot get it, get in touch with me and I can see what I can do. So when I started the study, I had to first obtain images of spiders and other insects. I ended up finding a quite large data set, and before I began the formal data processing pipeline, I decided to take a look into the data set to get a sense of what it contains and its potential. The rest of this talk will be focused on this process. My goal will be to show you how I use the Wolfram language to explore and make sense of a complex data set. And I hope that you can learn what you learned today can be applied to all sorts of other projects, not only biodiversity related. So the dataset I obtained is in a format called the Darwin Core Archive. This is a typical format for storing and sharing biodiversity data. That has the benefits of being efficient, in being flexible, and being stable. Some examples of these datasets are shown here with links to the datasets, including marine mammal abundance and distribution from the Cetacean Monitoring Service in the Eastern North Atlantic, a dataset with the environmental DNA sample from Monterey Bay, California, and the dataset used in this talk, which is citizen science observations, including photographs of jumping spiders across the world. And here is a link to the dataset if you want to download it and follow along this talk or explore it on your own later. So the Darwin Core Archive dataset that we will explore today was created by querying the Global Bioinformatics Information Facility, or GBIF, for all records from the taxon Salticidae. It's a family of jumping spiders, and from a source called iNaturalist research grade observations. So iNaturalist is a citizen science platform, and it has a ton of data, so that's why I chose this. And also I'm a regular user and contributor to the platform, so I was excited at the opportunity to be able to use a platform dedicated to citizen science. I'll be using the word observation a lot in this talk, and when I do, I will refer to something like this, which is a record of a living organism at a certain point in time. This will include some information about the organism, including its identification, and when it was observed and where it was observed. It can include one or more photos, and whenever there are two or more agreements with identification, it becomes what's called research grade and goes into these public data sets. So this query returned a data set with about 67 megabytes in size with more than 100,000 observations. So once we have this data set extracted into this path, we can take a quick look at the files that are inside. This shows us about 8 files. We won't look into all of these files today. Instead, we will focus on a few specific files which have the data that we're looking for. In this case, we're looking for a meta file, which describes the content of this archive, a core file, which in this case will be an occurrence format data file, since there can be different types of core files. This will contain identification and location information for each spider observation. It will also we'll also look into a file containing links to images of spiders for each observation. So right now we can start with looking into the XML files. A Darwin Core Archive will contain 2 of them. One metadata file, which contains metadata about the dataset itself, and a meta file with data about the file structure and content. With this, we can select all files with the XML extension. We see the first one is the metadata and the second one is the meta. And we're not focused on the metadata much, but it can be useful to take a peek at what's inside. Here I will use this import with the summary option, which will not import the whole data set. This is particularly useful if you're working with a file which you don't really know and how big it is, and it might tell you that it's too big to import into memory and you might want to try another approach. In this case, it's quite small, and we'll import the plain text just to take a quick look, and we can see some basic information about the download, the service, the query. But as I said, this isn't our focus, so we'll jump to the meta file. We'll try again using the summary. It's about the similar size. But now when we import the plain text, this is shows us suspiciously little. So I suspect it's more complex, and to really look at it, I will look at import it as an XML element. So I have to admit, I'm not particularly good at working with XML data, so please bear with me while we take a look inside. One thing I'll do right now, sorry about that, I will look at the tags of all XML elements with this, since an XML element contains 3 parts, and this shows us that the first element is an archive. So if I were to show you the whole XML element, it will look like this. So that's why I use the tags. Now we can look at the body of this element using this, and it shows us a list. We can do the same things we did before and do this to look at the tags, which tell us there's a core, extension, and extension elements, which probably reference these files. So I promise this won't be long, this XML data. So let's look at the first element again. Sorry. Yes. Here we see that the tag is a core file. We have in this attributes part, it's telling us here, it gives us a link to some documentation. It's telling us an occurrence. It's an occurrence type of file. And here we have a bunch of fields which refer to the columns with some documentation. Right, since we're just exploring this data set, I won't go more into this XML, the meta file, but when building a formal processing pipeline, this would be very useful, since it will tell us where the core file is, what each field means. So this is something that best to save for later. So let's look at the core archive. First, to do this, we need to find the core archive. To do this formally, like in a formal processing pipeline, we could use the meta file. But since we're not doing that right now, I'll just make a list of the files. Here, see that this is the occurrence data, and import it again as a summary. It takes a little bit. I'm importing, I'm importing it as a TSV, because beforehand in the meta file, I looked into it and I saw it's a TSV, even if it says it's a text file. So these things can happen. This data set has 250 columns. That's quite a lot, and more than 100,000 rows. But it's big enough, it's small enough to fit on memory. So I'll decide to import it as a tabular. Once I do that, I can see that I won't scroll through everything, but it looks like most fields are missing. This is probably because a Darwin Core Archive has a set of standardized fields, so we will have them even if most of that data is empty. We won't care about most of these fields, so the next step will be to simplify this data set. So let's do that. I have made a pre-selection of columns which have interesting data set. This includes the GBIF ID, which will be, it's an identifier for each observation. It will let us link later observations with images. Plus, we have data about the sex and life stage of the spiders, the time it was observed, where it was observed, and some identification, the genus and the species. I'll do this. I set the key columns as GBIF ID for merging later, and we have this simplified data set. Now we can scroll and see that there seems to be some interesting data there. So let's dive into this. The first thing that we can do with something like this is explore the diversity related in the diversity present in our data set. So the families Alticidae, the jumping spiders, include a few thousand species, and all within a few hundred genera. And because there's so many species, I will focus on listing the genera here. I do this, I use counts and I sort them so that the highest and the most common ones are first. But this still is a lot of text, probably more than 100 different genera. So we won't want to look at this right now. It's much easier to visualize it as a word cloud. I do this, it takes a second, and here we have this. We can see that we have quite a lot of genera, with most observations belonging to the genus Philippus. These jumping spiders are quite large and charismatic. They tend to follow you with their gaze when you pass by, so people tend to take a lot of these, a lot of pictures of these organisms when they see them. So to show you what I mean with charismatic, we can use the multimedia data in our data set to try extracting an image. To start, we need to find the multimedia file. Again, we could do this with the meta file, but since we're just exploring, we'll look at the files, see that it's there's this multimedia.txt file, import it, again, TSV, look at the summary. We see it has around 15 columns, 35,000 records, which means that probably many spiders do not have images associated to them, but still this is a good number to work with. And I'll import all of it. We can scroll, see that it has a GBIF ID, some data about the images, and the creator's analyses. Now, in this data set, there can be, each row corresponds to an image. Since one observation can have multiple images, this means that there can be multiple rows with the same GBIF ID corresponding to the same observation. So what I do before merging these two data sets is I can use aggregate rows. I aggregate them by GBIF ID, and to preserve all images, I wrap the identifier in this, so it's just a list. Also with license and the rights holder. And I can use join across to join these two data sets into something we can work with. Now, I want to be able to show you photos of what we have here. And to play it safe, I will select images with a CC0 license. And take a sample just to look at an example of what we have here. This is one record. We have here the images, and let's import them to see what we got. Sometimes there's many images, so I just put this up to 3 so that we get up to 3 images, although in this case it's just 2. And here we have a nice black spider, which happens to be a Phidippus audax spider. So previously I mentioned that Phidippus spiders are quite charismatic, and you just saw one, but I want to look specifically for one to try to convince you. So I can look at the first record with a CC0 license belonging to genus Phidippus, and I use select first and just extract the image locations. If I do this, I see that the paths in this server, and I can import them, grab them from this place and make an image collage. So here is a cute Phidippus spider. You can see that they have these large black eyes and these tufts of hair that look like ears or maybe little horns, and what looks like a mustache, but it's actually spaghetti pops. So, another thing that we can explore in this data set is the fact that jumping spiders tend to be sexually dimorphic. So that means that males and females can look different. So we can try looking for images of males and images of females and compare them. To import images, I wrote this short function, which just imports the first image for an observation, and I use this hack, which basically changes the this part of the URL to small, so that the import asks for a small version of the image, and that will help a lot in making imports much faster for this talk. So now I do this, and it takes it's pretty quick. And here we have on the left a male. They have usually they're more colorful. They have longer legs and slimmer body. And on the right you have a female. They tend to be more cryptic colored and more stout. This data set was obtained to propose a way of measuring mimicry by taking a neural network, training it with images, and the idea is that the neural network will be able to learn features of what a jumping spider looks like. But there's quite a big variety in their appearances, and before training our neural network or selecting our images, we might want to take a look at the diversity of images that we have in our data set. So we can do that with something like this. First, I'll select all the genera in our data set with CC0 licenses, and we have about 100. So that's quite a lot to run right now. And instead of running all of that, I will use a random sample to get 10 of them, and then make a image collage. I put this pause to avoid hitting the rate limit if there is one for the server. And here we have a nice sample of different spiders. You can see that sometimes the spider is actually not there. Like in this is probably its hide, like its little tent. This photo, it might be quite camouflaged, and maybe not all of these photos are great for my purpose, but this is actually a good result, because this informs us that there might be certain types of processing or curating that one will need to do in the formal data processing pipeline. Yeah. Our dataset also includes location, includes geographic information. So let's leave this aside for now and use the built-in geographic functionality to play around with our data. To start, I would typically look at what we have as our in our geographical data. We see that it's numbers. It looks like real numbers, decimal latitude and longitude. And what we can do to make them into a Wolfram language expression is wrap them in geopositions. So what I did was write this function. It will, if the values are there and they're valid, it will make a geoposition and wrap them in geoposition. And if not, it will return missing so that we can easily filter them out. I use transform columns with this function to add a column to this tabular object. And if we scroll to the end, we see that we have a geoposition column right here. So now let's use that to look at how spiders, or at least spiders in this dataset, are distributed all around the world. I remove the missing. I grab a random sample of 1,000 to avoid cluttering this image, this graphics, and use geographics. This shows us how the photographs observations are distributed. Have to take into account that this doesn't really tell the distribution of spiders. Actually, it's the distribution of mostly of the users of this platform of iNaturalist. But we can also zoom in if we want to, and zoom into a particular city or a country. So I come from Mexico City, and I'm curious about seeing what spiders I can find there. And to do that, we can first select records that are from Mexico. Use this, maybe make the next steps a bit faster, and we see we have around 4,000 observations. Then I can use geographics again, wrapping these positions in geomarker. And a neat way to select from a certain location is to use GeoRange. I use GeoRange Mexico City, add some padding, and wait a little bit, and then it will show me the distribution within this certain range. So if you're following with this code and you want to look at a different place, you can remove this. You can use freeform input with control equals. And if I put Mexico City, and have to think a little bit, and then it will give me this option of accepting. Then you can change the place to wherever you want. So now I have these locations, but I also want to look at pictures. Let's go back to our data set. And instead of looking pictures for all these spiders, I just want to get a representative image for each genre. So first I will do this count, like the counting and sorting them, as we did before. Since this is a smaller data set, a smaller location, and we have much less number of genera, and we see that the genus with most observations now is Paraphilippus, another genus, followed by Philippus. Now, I'll use the same code that I used earlier to get the images. This time I'll make a function for it, since I might want to use it later. And if I look at the top 10 most common spiders and make a collage, wait a little bit, we can see that this is what is most common in Mexico according to our data set. We see our friend here, Philippus, there, plus a few other species. So this is nice to know, but the abundance of spiders also changes with the seasons. Luckily, we have information about observation times, which we can use with the date-related functionality in the Wolfram language. And I'll show you an example. So I want to look at, again, observations in Mexico. And I look at the values in the column event date and select date objects. So thankfully, when importing this data set, the import automatically converted these values to date objects, which will make it much easier to work with. I use date histogram, bin it by months. I use date reduction year to remove the year specification, so we just see by months, and we get this histogram. We can see there's two peaks. There's second peak is in August. This coincides with the rainy season in many parts of Mexico, so that makes sense since rain means there will be more plants, and more plants means more insects, and more insects means more spiders. But so I will be visiting my family back in Mexico in December. And in my spider watching hikes with my family, I might want to go prepared and look at what I might expect to find. So to do that, I will select all spider genus from observations in December. Get this union to just get one per occurrence, and I get this list. And I can use the get images from genera function I wrote earlier, make an image collage, and just wait a few seconds while this downloads to see what I would expect to find when I go to Mexico. Yeah, and here we have it. You might recognize Philippus here. And we have and quite a nice range of other spiders. Probably there won't be as much since also it's colder, but it's nice to know that there will be jumping spiders when I go visit. So as this talk ends, I hope that you've enjoyed this presentation, and I want you to take home a few key points when you think of tackling a new data set. So I would suggest using the Wolfram language to quickly explore the new data set and use it to skim through the data to get a bird's eye view of what you will be working with. Aim for short code snippets with big results. If possible, use single functions. This will help you avoid getting stuck on details. Also, browse through the documentation, the Wolfram function repository, or even ask the notebook assistant for suggestions. And sometimes you have something very specific in mind, and you might find a function that does exactly what you want. Or if not, you might find functions that give you new ideas about what you could do with your data set, things that you might have not thought of before. And at last, keep note of what works and what doesn't. Right now, I didn't really talk about the paths that didn't work. And when I was exploring the jumping spider study data for the study, but knowing what doesn't work is also very useful when before you start committing to a dedicated processing pipeline. So again, thank you very much for listening, and I hope you enjoy the rest of the Wolfram Tech Conference. I will be looking at the Q&A, and 1 second. Okay, yeah. Tadeo is curious if this can be done for other organisms, such as with plants. A great presentation. Thank you very much. Yes, of course, there's basically any species that is in common enough to have observations, like people taking photos of them, will be on iNaturalist. And GBIF will import data from iNaturalist every once in a while. Okay, I'll answer another question. So what organizations provide their data in Darwin Core format? I think any organization that works with biodiversity related data will likely do that. I'm not really sure if it can be regional, like maybe some areas use a different format, but I think pretty much all I've found is in Darwin Core format. Although some smaller data sets might just focus on maybe TSV or CSVs without much structure. Yeah, well, I think the Stephen's talk is supposed to start now or very soon, so I think I'll leave this by now. You can reach me if you have any questions. And again, thank you very much. I hope you have a nice rest of the Wolfram Tech Conference.